So as Nicola already mentioned, I will be talking about data independent acquisition strategies and particularly how we can use that for high throughput applications in combination with the Evosep one. And the fact that many of you have joined this webinar now tells me that there is a great interest in getting higher throughput. And I just copied this figure here from the Evosep webpage, where it basically showcases the different methods and the different gradients that you can acquire with the Evosep 1 system, going from rather low throughput methods. Note that low throughput here means still 44-minute gradients, to very high throughput gradients like the 300 samples per day gradient, which is just about 3 minutes. And the other fact that I know from you is that you're not only interested in high speed, but you're also interested in high proteome coverage and data independent acquisition. So for the rest of the seminar, I will talk about why data independent acquisition is a good idea for short gradients. And I guess you've all seen many comparisons between data dependent acquisition and data independent acquisition. So just to cut it short, in classical data dependent acquisition, we would look at these all of our peptides eluting from the chromatographic column and measured by our mass spectrometer. So these are all the green signals that you see here in the background. And then in data dependent acquisition, the mass spectrometer would basically jump from one feature to another to acquire MS spectra of as many precursors as possible. So this is a sequential process where we go from one precursor to the next, to the next, to the next precursor. In contrast to this, in data independent acquisition, we acquire signals from many precursors in parallel. So instead of going from one to the next precursor, the mass spectrometer cycles through predefined ranges in the MOZ range, selects them with the quadrupole, and fragments all ions in parallel. And as we know from many areas in our life, parallelization is usually a good idea when we're going for higher throughput or higher speed. And so conceptually, this is why I think that DIA is actually a great fit to short gradients. So how does it work in more detail? Actually, DIA is not the newest idea. It dates back at least almost 20 years now. And one of the first publications showing this idea is this one from 2004 that actually explains already all the things that you need to know for this webinar today. So as mentioned before, in DIA, we will divide our entire mass range into small segments that we can then isolate with our quadrupole and fragment in a collision cell. So in this case, the width of these segments is about 10 Dalton. And then the mass spectrometer cycles through the entire mass range in these 10 Dalton steps. And this is done in a deterministic way, so that throughout the entire chromatographic elution time, the mass spectrometer will do the very same thing over and over again. Then interesting thing here is that we have two times here. One of them is the overall cycle time. In this case, it's TF. And the other one is the scan time. So that is the time we spend on a single DIA segment. So just keep this in mind for later on. In general, optimizing a DIA method for your application means that you would like to balance the identification with the quantification. And this is because typically identification gets harder and harder if your isolation width gets wider and wider. This is simply because the complexity of the MS2 spectrum increases. The more precursors, the more peptides are fragmented within a single DIA segment. At the same time, so for most identifications, you would probably choose as narrow mass ranges as possible. However, this would also increase your cycle time, meaning that if you look at this chromatographic peak here for a peptide, you will get less data points for your quantification. And this is typically the balance that you always have to take when doing data independent acquisition methods. And this is not new. There's a lot of great papers out there, and I can highly recommend some of these if you're interested in really squeezing everything out of your DAA methods. In this webinar, I will specifically talk about what actually happens when we now go for these higher and higher throughput methods. So basically what means going from a 120-minute gradient to a 20-minute gradient. To some extent, we're just squeezing our gradient together. So this means we will have more peptides co-eluting at the same time, and our chromatic peaks are going to get narrower and narrower. And today I will mostly talk about how we can address this on the mass spectrometry side, and Vadim in the next talk will talk about how this can be addressed by advances on the bioinformatics part. So hopefully together this will give you a very good overview of how you can tackle these challenges with modern DAA methods and data analysis. So to cut the long story short, having a longer cycle time means you have less data points across your peak. And if we squeeze together our gradients, this also means that our peaks will get narrower and narrower. So in other words, in order to get the same number of data points for your quantification with shorter gradients, you also need to speed up your cycle time. So instead of maybe having 3 seconds here with a longer gradient, to still get a nice chromatographic profile for your peptides, you would need to be already twice as fast if your peak width is only 15 seconds instead of 30 seconds. And there's many approaches to do this, and I think it's very good advice when you're starting with DAA to check this, what is kind of a good or optimal number of data points per peak for you to get accurate quantification. And in this paper from the diagnosis people, they had a very nice strategy looking in order to find the optimal point, not looking at the total number of identifications, but balance their quantification with the identification. So for instance, look at peaks or proteins quantified with enough precision, but also as many as possible. So this is, I think, a very good strategy to start optimizing DAA methods for short gradients. Something we are working on right now together with Thermo is, because we don't really want to give up the specificity of our narrow isolation windows, is to just speed up the acquisition time. So in this case, we use the new signal processing method that basically allows us to get the same mass spectral resolution in about half the time. So what this means for our DAA method is that we can still have rather fast DAA cycles with many windows, but now in a time that is short enough to also sample sufficiently very narrow peaks, for instance here just a few seconds with a cycle time of about 1 second. However, one problem with faster and faster acquisition cycles is also that you spend less and less time in each DAA segment. So this is shown here, where with a slower method, we still spend 32 milliseconds, for instance, in one DAA segment. And if we go faster, we spend only 60 milliseconds in that segment. So meaning that we actually have less signal now. So meaning that our mass spectrometer has to become more sensitive. And this is, of course, an extra challenge for the data acquisition system. So how could we solve this problem? And we've been thinking about this for quite a while, and we think at least one way to address this problem is to make use of ion mobility. And this is now the Bruker system that is equipped with this so-called trapped ion mobility device in the front part of the mass spectrometer. And a very interesting feature of this device is that it traps all the ions in parallel and then releases them from the TIMS device as a function of their ion mobility. So they're all stored and trapped in parallel, but released sequentially. And this is a very interesting feature. And particularly for DAA, when looking at this figure, now our data becomes three-dimensional. So we have data in M over Z dimension, we have data in retention time dimension, and in addition, we have this eye mobility separation. And if the technology now allows me to just switch my screen, and I hope you still see it, and we can actually browse into such a three-dimensional data set and look at how the peptides are distributed in there. So you could zoom into specific peptides of interest, and you see all of these features, they are peptides. And ideally, we would like to fragment all of them and get their fragmentation information for identification and for quantification. And a very interesting feature of this data space is that if we look in the, if we look in the M over Z dimension versus the eye mobility dimension, that's showing here, then we see this very nice correlation between mass and mobility. So just ignore this cloud over here. Those are singly charged precursors that we would like to exclude. But what we are interested in is here these peptides. So this is the big cloud of the peptides. And if you look at them in this dimension, you see a very clear correlation between mass to charge and the eye mobility. And looking at this for long enough, we thought, okay, maybe that's a very cool feature that we could also use for data independent acquisition. And this was the whole idea behind the DIA passive methods that Nikolai already mentioned before. So the idea here is to capture this ion cloud of peptides as it eludes from the TIMS device by synchronizing the quadrupole mass selection with the ion mobility separation. So our quadrupole would basically jump as the ions come out of the TIMS device. So it would start somewhere over here where this red ions elute, fragment these, and then within the same TIMS scan, also fragment the yellow ones, the green ones, and the blue ones. And this is kind of a time warp feature because remember, we initially stored all the features in parallel and we sequentially released them. So we do not waste ions because we are still looking at all of them. We're still capturing most of the signal. And I think this is a very, very interesting feature for data independent acquisition. However, it also makes it a bit more complicated because our problem of finding optimal window placements now is not only like a two-dimensional or even one-dimensional problem where we just need to find the optimal distribution of our windows in the M over Z dimension, but now we also have to consider the eye mobility dimension. So instead of having windows only in M over Z, we're now talking about boxes. And the challenge is to place these boxes in a way that we have enough specificity, that we have enough quantitative accuracy and enough data points over the chromatographic peak, and that we do not give up our sensitivity that we have intrinsically in the passive acquisition mode. So it's basically a whole new dimension for method optimization. And we try to formalize it in a way that we mostly try to conceptualize our method designs in terms of how we place the windows, then the overlap of these windows, for instance, in M over Z or eye mobility dimension, then how many steps or how many windows we would like to acquire per DIA passive scan, then how many of these DIA passive scans we would like to do in an acquisition cycle. So you could have rather broad M over Z windows and actually cover all precursors in a single DIA passive scan. Or if you would like to work with more narrow M over Z windows, then you could split up the entire mass range to multiple DIA passive scans. And this is then how this looks in reality, where you could load one of your 21-minute Evosep runs and then overlay your window placement on top of a two-dimensional map, eye mobility versus M over Z. And then the idea is to set this acquisition area so that you cover most or even all of your precursors that you're interested in. And Patricia Skowronek was a very talented master's student and now a PhD student in Matthias's lab was brave enough to optimize these methods, and she was looking at all different parameters that you could only think of. And one of them was, for instance, the width of the isolation window. And you can nicely appreciate the effect that this has on the number of identified precursors, for instance, but also on your quantitative accuracy. So meaning that if you have rather wide windows, you can be very accurate. You have very low CVs, but this also comes at a cost of proteome coverage. And turning the other way around, if you have rather narrow isolation windows, you're getting better in terms of identifications, but you're losing a bit on quantitative accuracy. So for us, it turned out the kind of a sweet spot is somewhere in the middle, where we have 25 Thomson isolation windows, which gives us still a decent quantitative accuracy with CVs somewhere around 10% or even below 10%. So in the beginning, I showed this figure from the Evosep website, basically saying that you would have to you have the choice going from high throughput to high proteome coverage. And my goal was to convince you that nowadays you can actually get both. So using one of these optimized CIA passive methods shown before allows you to now accurately quantify over 4,000 precursors per minute, somewhere in the middle of the 21-minute gradient, which then turns into over 6,000 quantified proteins, which I think is something we would have only dreamed of just a few years ago. And with this, I would like to thank the many people involved in this work, my new lab at the Jena University Hospital, the old lab led by Matthias Mann, where still most of this work was actually performed, and all the collaboration partners for very fruitful collaborations throughout the years. Thank you for your attention.